Hi, my name is Matteo and I'm a lecturer officer at MBSA.
I'm Liza, I'm the Secretary General of MBSA.
Hi, I'm Desiree and I am the Vice President of MBSA.
Hi, I'm Bernice and I'm a second-year student of medical biochemistry. Hi, I'm Zafra and I'm a second-year medical biochemistry student.
Hi, I'm Andy and I'm the President of MBSA. And welcome to the MBSA at Science in the City.
We are MBSA, and we are a student organization that represents the students following the medical biochemistry course, a course that focuses on medical research. We are currently in the middle of a global pandemic, and because of that, I'm sure that most of you watching has been swabbed at least once or twice in these past months. But have you ever thought of what happens once your sample is taken? What goes on behind the scenes? Well, stay tuned to find out.
As Andy said, today we will be breaking down and explaining the process of COVID-19 testing and highlighting what is DNA, what is PCR and how it is used to test for the virus and other testing methods.
DNA is basically an instruction manual which determines how your body looks and works. This means that it contains the instructions for all the processes which need to occur inside your body. For example, in order for your nails to grow, the cells in your fingers need to follow all the instructions found in the DNA. This means that everything that makes you you and that makes you different from every other thing in the universe is your DNA. Every living organism has DNA. This includes viruses. DNA is double-stranded and looks like a twisted ladder. It is made up of small building blocks called nucleotides, which each contain a sugar. There are 4 different types of nucleotides in DNA. These are adenine, thymine, guanine, and cytosine. In DNA, adenine always binds to thymine, and guanine always binds to cytosine. Since DNA is double-stranded, if on one strand we have adenine, exactly opposite it on the other strand you would have thymine. If you have cytosine on one strand, exactly opposite on the other strand you would have guanine.
The process of DNA replication starts with having one double strand of DNA. Like Sephora explained, double strand of DNA will look like a twisted ladder. Now, this double strand will open up to form 2 single strands of DNA. So, like having a ladder which is twisted and it will become like a normal ladder and it will open up. Now, these 2 single strands will be instructed to double so that another 2 strands of single-stranded DNA will form. Thus, overall, you will have 4 single strands of DNA. These 4 single strands of DNA will then pair up to form 2 double-stranded DNA molecules. Overall, the result of DNA replication would be that one double strand of DNA will be doubled into 2 double-stranded DNA molecules.
When you do a swab test, the doctor collects a sample from your nose or your throat and sends it to the lab, where they analyze it to see if there is viral DNA present in your sample. If viral DNA is present, this would mean that you have a positive result. If viral DNA is not present, this would mean that you have a negative result.
What is the difference between viral DNA and human DNA?
COVID-19 is a type of coronavirus. This basically means that the outside of the virus looks like a crown. Coronaviruses have RNA instead of DNA. The main difference between RNA and DNA is that RNA is single-stranded, while DNA is double-stranded. In RNA, the nucleotide building blocks have a different sugar than that found in the DNA building blocks. In RNA, we also have one different type of nucleotide. Instead of thymine, we have a uracil. This means that the building blocks of RNA are adenine, guanine, cytosine, and uracil.
The polymerase chain reaction, otherwise known as PCR, is a procedure used to amplify the amount of DNA within a sample, meaning to multiply it. We use PCR to detect the presence of DNA and also to analyze it. The swab test works using PCR. The first step is to take the sampled swab and place it into a test tube, which is heated to a certain temperature. This step is done so that any DNA found within the sample will be broken down into the two individual strands, meaning that we unzip the double helix. The next step is to lower the temperature of the test tube so that we can add the nucleotides. These are the building blocks of DNA. The nucleotides will bind to each individual strand and form a complementary strand, meaning that we took one double strand, split it into two, and now from those two individual strands we've generated two new double strands. This means that we have double the amount of DNA that we started with. This is what PCR does. It is an exponential reaction, meaning that with each individual stage we double the amount of DNA we started with.
Why is it important to multiply DNA?
The DNA in the sample is multiplied, as having multiple copies of the target DNA makes it easier to find and identify. This is the difference between having to find one dot on a large screen versus having to find 100. A fluorescent dye is added to the sample. This dye attaches to the viral RNA like a gift tag, and thus, if viral RNA is present, the sample glows, giving a positive result.
A rapid test, the same thing.
Rapid tests are another form of diagnostic test, meaning like the PCR tests mentioned previously, they can be used to diagnose a current coronavirus infection. However, unlike the PCR tests, which test for viral DNA, rapid tests, also known as antigen tests, are used to test for viral proteins. Antigen tests are also called rapid tests because the results can be delivered within minutes.
Are there any other testing methods?
That's a really good question. There is a third test, known as the antibody test. However, it is not diagnostic, which means it cannot diagnose you if you currently have COVID-19. It can only check whether you've had it in the past. This test is carried out by taking a blood sample and takes anywhere from 1 to 3 days to come back. The antibody test works by, as the name suggests, testing your levels of antibodies. Antibodies are something that your immune system creates once you've been exposed to a specific virus, and it helps you fight off the virus the next time you encounter it. However, your body may take up to weeks to create enough antibodies needed for a positive antibody test for COVID.
Are the tests even reliable?
The PCR tests are the most accurate of all the testing methods. The antigen tests are more prone to give false negative results, meaning they are more likely to miss a current coronavirus infection. Despite the PCR test being the most reliable, no test is 100% accurate due to human error.
Thank you for joining all of us today. Hope it was informative. And if you have any questions, don't hesitate to contact us on our Facebook or even on our Instagram. Stay safe. Bye!